I just want to introduce next speaker, Dr. Ying. She's a senior research investigator at the University of Pennsylvania, and she's going to be speaking on CRTH2 PTGDR2 antagonist reversing inhibition caused by the elevated PTGDR2.
Good morning. I'd like to thank the meeting organizer for giving me this opportunity to present our work here. This work was conducted under sponsored research agreement between UPenn and Kaisara, now part of Antagonists. Previously in our lab, we discussed et al. Showed that prostaglandin B2 as well as the synthesis PTGDS are elevated in bulk scale of men with ADA, and this is in the mouse when you apply exogenous PTG2, it inhibited hair growth. And in human hair follicle organ culture, PTG2 also inhibited hair growth compared to control. PTG2 combines to two receptors. One is PTGDR1, which is the DP1 receptor. The other is DP2, also called CRTH2 or PTGDR2 receptor. This made an important finding that in the mouse that knock out the PTGDR1, when you apply the exogenous PTG2, you still see this hair follicle growth inhibitory effect. However, if you knock out DP2 with the PTGDR44 receptor, when you apply PTG2, then you don't see the hair growth inhibitory effect anymore. So this means that PTG2 inhibits hair growth through its receptor DP2. So our question is, can DP2 antagonists prevent or reverse the PTG2-mediated inhibition of hair growth? We first showed that DP2 is widely expressed in human hair follicle keratinocytes and sebaceous gland. This is in the bulk scale. It seemed to be elevated because these pictures were taken under the same setting. In the literature, it also shows that DP2 is expressed in dermal papilla. However, with this particular antibody we're using, we seem to see minimal expression in the DP. And same holds true with the mouse skin and hair follicle. And we tested PTG2 inhibit hair growth in the Philpott organ culture model. We cultured the human follicle for 7 days with a media change every 48 hours. And we found that at 10 to 20 micromole PTG2, it inhibits about 40% of hair growth compared to control. And it also induced a pathogen-like morphology in the PTG2-treated hair follicles. We showed that with increased concentration of PTG2, you have a decreased cell proliferation as shown by the Ki67 positive cells, and with increased apoptotic cells. We tested several DP2 antagonists. The one that I'm going to talk about here today is AM211. It's a potent and selective small molecule DP2 antagonist. Zheng et al. Showed that at about 10 nanomole, AM211 can inhibit about 60-70% of PTG2 binding. We first tested AM211 in hair follicles from both male and female donors. That we found that both male and female hair follicles in organ culture can be inhibited by PTG2. And when added the antagonist at about 10 nanomole concentration, it can restore the hair follicle growth to about 80% of the control. However, we found at a higher concentration, such as at 100 nanomole, this protective effect seemed to disappear. And then we did a finer dose curve of AM211 with reduced gas between the concentration. Again, we found that the most effective dose seemed to be between 10 and 20 nanomole. At this point, the compound that we're using, the AM211, is about 95% pure. So we wonder if there's any impurity in the compound that can actually be detrimental to hair growth at high concentration. So we made another batch of AM211. At this time, it's at 99.5% pure, which is equivalent to the clinical grade of AM211. So we're happy to see that with the purified version, AM211 does not exhibit the growth inhibitory effect anymore at high dose, even as high as 1,000 mmol. And this slide also shows with the addition of the antagonist, it restores the proliferation of the hair follicle and reduces apoptosis. These are the quantification of the number of proliferation and particles. And previously in our study, we showed that the endogenous PTGDR2 level peaks during pathogen. So we asked the question that if you gave exogenous PTGDR2, say in the middle antigen, can that shortens antigen and lead to premature entrance to pathogen? And the following question is that if you gave the antagonist during the antigen prior to the peak of the endogenous PTGDR2 level, can that extend antigen and delay onset of pathogen? So in the first experiment, we topically treated the mice at postnatal day 21 antigen and all the way to target at postnatal day 42. We showed that in the control group at each time point, the hair follicles are still mainly in late antigen. However, in the PTGDR2 treated group, at day 39, they are mostly in early antigen, and at day 42, they further advance to the pathogen. And we also showed that there's significant decrease in the number of KI67 positive cells in the treated group, which is consistent with what we saw in the hair follicle organ culture. And in the next experiment, we depilated mice and we started the antagonist treatment at day 8 in the middle of antigen, and we harvested the skin at day 22 and it started skin. And as you can see in the control, most of the hair follicles are in pathogen. However, in the antagonist treated group, you can see these areas of the hair follicle that's not in keratine. And then we did the hair follicle stage quantification again. As you can see, there are reduced percentage of hair follicles in keratine in the treated group. And we plucked the hair from the mice and reviewed the hair supplement. And we saw that at least first two doses that there's significant increase in the hair supplement in the treated mice. We do see this decrease in hair supplement at higher dose. Just remind you that in this particular experiment, we were still using that 95% pure antagonist. So I think that's similar to what we have in the other project system. There is some impurity at high dose that activates. So taking all these data together shows that antagonist of AM201 extends the phase of antigen and leads to longer hair track. I'm just here to talk about a different step of the experiment. In our testing of the antagonist, we actually went through a lot of donor particles. That we found that some individuals are pretty sensitive to the inhibitory effect of PTGDR2, while some other individuals seem to be pretty resistant to the PTGDR2 inhibitory effect in donor particles. So we wonder if there are any SNPs in the PTGDR2 gene that are associated with hair follicle sensitivity to PTGDR2. We sequenced the entire PP2 gene plus 2 kb upstream and 1 kb downstream. Initially, we started with 35 samples. Among the 35, 21 of them are PP2 sensitive and 14 are not sensitive. We actually identified 3 SNPs that are predominantly present in the PP2 sensitive group. These are the location of the 3 SNPs in the gene. And the 2 pink SNPs are kind of like our negative control because they're within the region of the 3 blue ones, but they're not associated with the sensitivity. The Fisher's exact test, our p-value at this time is 0.04. So to further confirm these findings, we did plasma SNP analysis of 65 additional new samples. And it supports our initial finding by iron current sequencing that these three SNPs are actually associated with PTGDR2 sensitive group, and our p-value at this point is less than 0.01. And we try to understand what is the impact of these SNPs on the function of the BP2 gene. So in the literature, this one report suggests that while the SNP is associated with increased mRNA stability of the BP2 gene, this is a report by Wang et al., and this is the in vitro transcription system where we make construct contains the SNP, and the mRNA is more stable compared to the wild type allele. So we did do PCR analysis of the BP2 gene in donors with or without the SNP. We found that in the donors with the SNP, it seems to be a fourfold higher level of BP2 gene compared to the wild type. However, the p-value is not significant at this point due to large variation among individuals. To summarize, PP2 antagonists A and P211 reverse the hair growth, inhibit rate-backed phosphate PTGDR2 in human hair follicle organ culture, and exogenous PTGDR2 in mice cause premature entrance into pathogens, and the PP2 antagonists extend and lead longer hair span. We also identified three SNPs in the BP2 gene that are associated with sensitivity to PP2 inhibition. These SNPs may serve as markers to identify patients with higher risk of PTGDR2 attack. I'd like to dedicate this talk to my dear colleague and friend, Dr. Zheng, who unexpectedly passed away about 2 months ago. He was the major contributor to every single experiment of this project, and we listened very much, and we are trying to set up a neurofund to benefit from a lot of this. Many in the lab that helped with animal works and downloads of SNP analysis, and each in Adam Dyson helped with the follicle effect. Dave and Harini and Dr. O'Brien helped us with the iron current sequencing. Our wonderful collaborator Serge and Sarah helped with the hair follicle stage analysis. Last but not least, I'd like to thank Dr. Ken from Cross-Sigment and his staff and to the field for time as we started human cell tissue for hair follicle organ culture. Without them, this work cannot be done. So thank you very much.
So really interesting and novel talk. I think we've got time for one question. If there's anyone in the audience with a question?
So in the mouse studies, you got about a 2% increase in length that consistent with just delaying pathogen. Do you know if the drug's only active late in or is it transition?
We find whether it promotes the activity of anything. That in fact is not very obvious.
And does it have any effect on growth other than at the very end of the cycle?
The growth rate.
Well, growth rate or just the net effect is a small effect that's consistent with that slight increase in the duration.
Right. We think it's the prolonged end phase that leads to the longer hair span.
So for it to be active, you really have to hit that transitional phase.
Right.
That's awesome.
Do you know how long you can safely use a topical corticosteroids on the scalp, and does it cause pain of the skin? And then I have a second question.
I think that question for the speaker. Yeah. We might save that question till later.
Dr. Zheng.
Yeah, we'll save that till later.